Today we're going to talk about microbial metabolisms. We're going to talk about how these different molecules and microbes are going to be able to use complex controlled biochemical reactions in order to get the energy that they need and in order to help them to reproduce. So with metabolism, that's going to be the sum of complex controlled biochemical reactions within an organism. Its ultimate function is going to be to help reproduce that organism. So it's going to be divided into two further types of reactions. We have what's known as catabolism, and that's going to be the breakdown of larger molecules into smaller products. We have exergonic, which means it releases energy. And so that's going to help to release energy from whatever food or whatever molecule it's taking in to process it or to fuel it. Anabolism is going to synthesize large molecules like macromolecules from smaller products. So it's going to take what that cell has after it breaks things down and it's going to use it to make the things that that cell needs. So it helps it grow and then ultimately will help it divide and reproduce. This is going to be an endergonic reaction. So that means it's going to require energy. So catabolism, exergonic, releases energy. Anabolism is going to be endergonic, and that requires energy. If you want to think about it kind of like some building blocks, if we, for example, as a human, take in let's say a piece of chicken or a piece of steak, we're going to take that in, our body's going to break that down into smaller building blocks. You can think of those as the sandwich or the steak or the chicken. Think of it as being built with Legos. Those Legos then will be broken down, that would be catabolism, and then it's going to release energy when it's breaking those bonds between the Legos. And now you have all these different molecules or pieces that then you can then reuse to rebuild something. So when you're rebuilding, it's going to be anabolism. So you take those blocks and you rebuild it into things that the cell needs. And so that is requiring energy to build the things that that cell needs or make larger molecules, and that's your anabolism. Now with this, we're going to have what's called oxidation and reduction reactions. These are going to involve the transfer of electrons. So if it's oxidation or oxidative, you're going to have an electron, which is shown here by the E, that is going to be removed from the molecule. And when it does that, that's going to release energy. And so that would be exergonic. A reduction is going to be the addition of an electron molecule. And so that's going to store energy. And so with that one, that you can see goes into the blue molecule. So let's look at these. We have the orangey peach. It's going to be releasing that molecule there. And so when it releases that molecule, it's going through oxidation. It transfers that molecule over to an electron acceptor. And so that blue is going to actually take it on. And so that will be a reduction when it takes it on. So it goes from not having an electron to having an electron. So it's reduction. Electron to not having one, oxidation. So this is always going to occur simultaneously because when the molecule loses the electron, it's then going to have to be transferred to another molecule. So the electron carrier molecules that we're going to talk about in the future slides coming up are going to have three different types. We're going to talk about a nicotinamide adenine dinucleotide, and so that's always called NAD+. You have the nicotinamide adenine dinucleotide phosphate, and that's NADP+. And then you have the flavin adenine dinucleotide, and that's the FAD. So we're going to look at these coming up. So with ATP production and energy storage here, that's going to be your major energy carrier of the cell, that ATP. So that's going to be like the battery of the cell. And of course, that's made at the powerhouse of the cell, which you should know is going to be your mitochondria. So with this, it's going to be concentrated and stored in high energy phosphate bonds. And so looking here, you have your adenosine triphosphate, that's the ATP, that's going to be on your left-hand side. Notice how it has 3 phosphates that are hooked together. Well, the last phosphate between phosphate 2 and 3, you see where the star is, that's where that energy is going to be stored. So when you break that bond, you release that phosphate, it's also going to release a packet of free energy. And so then it becomes ADP, adenosine diphosphate, and you have that free-floating inorganic phosphate and some energy that's going to be released there. So this is going to be used for most metabolic processes that require energy. So with this ATP production, you're going to have a substrate level phosphorylation. And so the phosphate, like we said, it's going to be transferred from another organic phosphorylated molecule. And so basically that is oxidative phosphorylation. So it attaches the inorganic phosphate using energy released from respiration. So with that photophosphorylation, it's going to attach inorganic phosphates using energy obtained from light. And so that would be through like photosynthesis. And so you can get it from food, or you can get it from food which got it from photosynthesis. So that would be the difference between like your autotrophs and your heterotrophs that you take in and use for energy, or other molecules do. So with these enzymatic roles in metabolism, you're going to have organic protein catalysts that are going to increase the rate of chemical reactions. Naming and classifying enzymes is going to be important. They're going to be named for their substrates. And so molecule enzymes are going to act upon specific things, and so that's going to basically be how they get their name. Generally, they will end in ACE. So if you see ASE, that will be an enzyme. So enzymes are going to be classified to types of reactions that they will catalyze. You can have what's called a hydrolase. Those hydrolases will catalyze hydrolysis reactions. That means adding water to the reaction, so hydro, water. Isomerases, got tongue-tied for a minute, they will catalyze and rearrange the functional group within a molecule to convert that substrate into a different isometric form. You have ligases. They will catalyze the condensation or joining of two molecules. You have lyases. These will catalyze and split the molecules with water. You have oxidoreductases. These will catalyze reactions and transfer oxidation and reduction reactions. And so we just looked at some of those with those molecules, how it worked. And then you'll have what's called your transferases. These will catalyze and transfer a functional group from one molecule to another. Now, enzyme makeup. So some enzymes are going to be completely made of proteins. Some enzymes are going to be made of proteins and of non-proteins. So you have a apoenzyme, that's an inactive protein. It's going to be bound to a cofactor, that's a non-protein substrate. So here you see the protein, here you see a coenzyme, that's an organic cofactor there. With this, it can bind to inorganic ions. These are going to be things like iron, magnesium, zinc, copper even, and can be organic molecules. So these coenzymes can be like a vitamin-derived molecule. And then as you put these two things together, they become a holoenzyme. So that puts the apoenzyme together and the cofactor together. And once they come together, they become active. And that makes it a holoenzyme. Enzyme activity is going to be important because enzymes will lower the activation energy that's needed or required for a reaction to occur. This can happen in four main steps. So you have an enzyme substrate complex. What happens there is that enzyme is going to interact with the substrate, the enzyme, and the substrate specificity. That is going to have an active site. It's going to have the enzyme where it's complementary to the shape of that substrate. So it has to fit perfectly. Two models are going to be regarded for this. You have one that's called a lock and key theory or a lock and key model. This is going to describe kind of a rigid enzyme that's going to have an active site that is going to bind to specific shapes. So if the shape is off at all, it will not bind. So it's going to fail to take into account protein conformation changes and accommodate a substrate molecule there, so it has to fit perfectly in. You can also have what's called an induced fit model. That's going to describe a more flexible active site that will kind of approximate the shape of the substrate, and that's going to mold to it. So you see that active site in the second picture that's coming in, and it's molding to the products or the substrates that are coming in. And so then that will help to produce that perfect fit as it molds in. So one of them is kind of a rigid structure, has to fit perfectly. The induced fit is where it's going to be able to come in and it's going to mold perfectly to those substrates. So what you see there, A is going to be your active site, or that A1, and then B and B1, B2, those are your substrates that are coming in. So those are the things that need to be able to fit into that enzyme's active site. And so here we have this transition state, so that shape of the substrate could be altered into an intermediate form. The substrate has features of both substrate and the final product. We have product formation, where that substrate is going to be converted into a product, and then we have product release. And so with this, that product is going to be released from that enzyme, leaving the enzyme completely unchanged. And so that has your 4 steps here. So we have enzyme substrate complex. We have a transition state where it changes or fits in perfectly. We're going to have that product formation. So once it comes in, fits in, let's see it break that bond, it's making that product, and then you'll see it in picture E releasing that product there. So there's going to be different factors that will affect how this enzyme activity works. So let's go back here and look at this. Let's say that the enzyme is going to be lactase and the product that's coming in is going to be lactose. So you had some ice cream, your body needs to be able to break down the sugar in order to use those glucose molecules. So it comes in, it fits in, it starts to bend that bond that you see in C, the transition state. It breaks the bond. Now we have 2 separate molecules, so we see that in picture D with the enzyme product, and then it's going to release that product. Now that's going to be usable forms of sugar for the body, whereas before in picture A, it could not use that form as you took it in. And so there's some things that are going to affect whether these enzymes work or not. And so the first one we're going to look at here is temperature. Enzymes function best at 37 degrees Celsius. That's going to be normal body temperature. Enzymes are going to remain stable at temperatures lower than 37 degrees Celsius, and they become denatured at a temperature that's going to be higher than 37 degrees Celsius. So when you denature a protein, that means to change its shape. You change its shape, you're going to change its function. So temperature can change its shape, which then will make it non-functional. So anything higher than 37 degrees Celsius will render that enzyme non-functional. pH is going to be considered. That will actually affect how the enzyme works too. So enzymes are going to function best at a pH of about 7. Solutions that are going to be either too acidic or too basic are going to cause that enzyme to become denatured, and then that's going to cease the function of that enzyme, so it no longer works. So substrate concentration increases, that can increase enzymatic activity, and enzyme concentration increases can also increase that enzymatic activity. And so you can see that there. With light can actually activate or inactivate some enzymes. So enzymes involved in DNA repair and pigment production are going to be activated by light. So that's another thing if it's going to be a light sensitive enzyme. So here is just a picture of denaturation. And so the normal functional protein is going to be on the left. Once it becomes denatured, that's what you see on the right. So remember, first thing, you have your primary structure. It's put together by amino acids and peptide bonds. Then it will corkscrew or zigzag. Then it's going to take those and it will bend them, and then it will hook several of those together. So we have different colors for the ones that are hooked together. And so if you denature it, you're taking away that shape and how it is put together. And so it's no longer going to perform the correct function. So that's your denaturation, or if you denature a protein. So with our non-competitive inhibitors or reversible inhibitors, these are going to bind at some place other than the active site. These are going to be called allosteric sites. So this is going to lead to a change in shape at the active site. So an inhibitor is going to be bound by a weak interaction. That will allow for that inhibitor to be easily removed from the active site, and then it will provide an opportunity for the substrate to come in and bind there. Now you can have what's called feedback inhibition or a negative feedback product that's going in there. That is going to be a way in which cells can control an action of an enzyme. So this will occur when that final product of a series of reactions is allosteric inhibitors of some previous step in a series. And once it accumulates enough of that enzyme there, then it's going to send a feedback loop back and tell it to stop producing this. We don't need any more of this. Now, going into carbohydrate catabolism. So we're breaking down carbohydrates as they come into this organism. So carbohydrate metabolism, we said, is a breakdown of the carbohydrate molecules, and we're going to use these to actually produce energy or ATP for the cell. The most common carbohydrate source is going to be glucose, and so that is going to be a simple sugar, a monosaccharide. That is going to be the central player in metabolism. So we're going to have two types of carbohydrate metabolism. The first one, cellular respiration, and then the second one, fermentation. Both of these processes are going to share a process referred to as glycolysis. Now, glycolysis is going to involve the splitting of that glucose molecule in a series of 10 steps, or catabolism, with the addition of 2 ATP molecules. So glycolysis requires energy to even start. So you see that glucose molecule there, you add in the 2 ATP, and that's going to start off that reaction. That is going to break that glucose molecule into 2 parts, and it's going to make it an unstable molecule as it's broken apart. So you split the glucose, and when you split that glucose, that is going to give you 4 ATPs, but you're only able to use 2 of them. So it's 2 usable ATPs because 2 of those have to go back in and start the next glycolysis reaction. So basically it gives you 2 molecules of pyruvic acid. That's what happens when you split that glucose. You get a net gain of 2 ATP because remember it's 4 total, but 2 have to go back and start the next glycolysis. And then you'll get 2 NADH molecules. And we just talked about that. That's your high energy electron molecules. And so this is going to be represented in our format that you see down there. So you see it starts off, we have 2 ATP that have to go in and start this reaction. So you do spend energy, break glucose, get 2 pyruvates, which are going to be those 2 little green molecules there. You get 4 ATPs, but 2 have to go back, so you're only going to get 2 net ATPs. And then you'll get the NADH, which is the high energy electron carrier. And during this process, you'll see that you do release water. Now coming into the next step. Let me go back to glycolysis, talking about it. This usually occurs if it's in a eukaryotic cell. It will occur in the cytoplasm, but with prokaryotic cells, it occurs in the cytoplasm as well. Cellular respiration is going to be a multi-step process that allows the organism to completely catabolize that glucose into carbon dioxide. Also, this is going to be known as aerobic respiration because oxygen is going to be the final electron acceptor. And that's going to be represented by using the following chemical equation. So I'm going to write that up for you. So you're going to have your glucose molecule, which is C6H12O6, and you're going to take in 6 oxygens. So you'll have the oxygen there, so we put 6 in front of it. O with a small subscript 2 is going to be oxygen. So those are what's going into it. So those are our reactants. Then you would draw an arrow. Sorry, I don't have enough room for this. And then you end up with a product of 6 CO2s, so 6 carbon dioxides, so that's a 2. You have 6 waters, 6 H2Os, and then the release of ATP or energy. I'll put an E for energy. And so this is what's being produced. This is what's going into it. So that's your reactant. So that's how you would write it out. I know it's a little bit messy. Sorry for that. I'm writing with my finger. So that's how you would write it out as a chemical reaction. So it's going to involve 3 main stages. You're going to have your synthesis of acetyl-CoA, you have your Krebs cycle, and then the final step, which makes the most ATPs, is going to be the electron transport chain. So let's start off with our first step, the synthesis of acetyl-CoA. So you have a preparatory reaction, and then that's going to be formed when two carbons from pyruvic acid are going to join coenzyme A. So you see your pyruvate, you see what that molecule looks like. It's going to join in with coenzyme A. You'll get 2 molecules of acetyl-CoA, 2 molecules of carbon dioxide, and then 2 molecules of NADH are going to be produced. That's your high energy electron carrier. And so you see, as we're making this carbon dioxide, that's what you're going to end up breathing out. Next, we'll go into the Krebs cycle. This is going to be a cyclic metabolic pathway that's going to allow these organisms to decarboxylate organic acids and then release the potential energy that's stored within them. So it's a series of 8 enzymatic steps that transfer energy and electrons from acetyl-CoA to coenzymes NAD+ and FAD. So here we get 2 molecules of ATP. You're going to end up with 6 molecules of NADH, and then 4 molecules of carbon dioxide and 2 molecules of FADH2. And so that's going to be your end products here. With the Krebs cycle, it does go around the cycle twice in order to get those products. Now the last step, which is going to be the most tedious step, but it's going to yield the most energy. This is your electron transport chain. It's going to involve a series of membrane-bound proteins that are going to pick up electrons and hydrogen proteins from the coenzymes that are NADH and FADH2. It's going to pass them from one molecule to the next through a series of oxidation and reduction reactions until they are going to be picked up by a final electron acceptor. Often it's going to be a molecule of oxygen. And so you're going to have 4 categories of those carrier molecules here. You have flavoproteins. These are membrane proteins which contain flavin derived from riboflavin or B12. That's going to be the initial carrier molecule of electron transport chains. You have what's called ubiquinones. These are going to be your lipid-soluble, non-protein carriers that are derived from vitamin K. You'll then have also metal-containing proteins. That's your third type. These are going to be the integral proteins that contain metal atoms such as iron, sulfur, and copper. And then you have your cytochromes. These are going to be integral proteins that are going to be associated with the heme molecule. So it's going to contain iron, non-protein substances, and it's going to be the molecule that's going to be found in blood. So heme is going to help to make up hemoglobin. You'll also have chemiosmosis. This is going to be a mechanism in which the flow of ions down the electrochemical gradient across this membrane are going to be used to synthesize or to make ATP. So energy is going to be released during the redox reactions of electron transport, and it's going to be used to pump proteins across the membrane, creating a proton gradient. And so these protons are going to be low down the electrochemical gradient through the protein channels called ATPases. And so with that, they're going to help to synthesize ATP in an oxidative phosphorylation and photophosphorylation process. So at the end of this, we end up with around 34 ATP molecules from 1 molecule of glucose. So the total yield from aerobic respiration, remember that's the one we just looked at, it requires energy. Glycolysis gave you 2 usable ATP. Remember you got 4, but you had to send 2 back to start the next one. So you get 2 usable. Krebs cycle gives you 2 more usable ATPs. And then the electron transport chain gives you around 34. Now this could be 32 to 34. So total maximum amount that you could end up with 1 molecule of glucose will be 38 ATP. Or they say sometimes anywhere from like 36 to 38. Now, alternative pathways for glycolysis can come in, and this would be like a pentose phosphate pathway. It can be used for the production of precursors of metabolites that are going to be used in anabolic reactions. These will help to make nucleotides for nucleic acids. It helps to make amino acids, and it can help make actual glucose. So this is going to net a single molecule of ATP. So not a lot of energy there. Or you can enter in to what's called this Intner-Dulerdorf pathway, and that's going to be a series of reactions that catabolize glucose to pyruvic acid. And so that pyruvic acid is going to use a different enzyme than those that are used in glycolysis or into pentose phosphate. That's going to give you a single molecule of ATP, a single molecule of NADPH, and it's going to have precursor metabolites. Another way to get energy is going to be a way that you would go to if you did not have oxygen available. That's going to be fermentation. So this is an anaerobic decomposition of organic compounds. Usually it's going to be carbohydrates, and it involves an organic compound, usually the pyruvic acid, as a final electron acceptor. Now with this, it's going to be essential for the function of like a generation of NAD pluses for glycolysis in order for ADP molecules to be phosphorylated into ATP. So this is going to be important in making those ATP. You're going to have two common fermentation processes. And so the first one is lactic acid fermentation. You go through this in your own body. If you're exercising or running, lifting weights, this is when your muscles start to burn. You feel the burn, and then it leads to muscle fatigue. So what's happening is, as those muscles run out of oxygen because you're working very hard, you're starting to breathe heavy, you just can't replace that oxygen fast enough. So you have a buildup of lactic acid between those muscle cells. That's going to create a burning feeling or a burning sensation, which also leads to that muscle fatigue over time. And so what's happening here is the NADH is going to reduce pyruvic acid from glycolysis to form that lactic acid. Another way, you could have alcoholic fermentation. So you'll have your pyruvic acid undergoing decarboxylation, where CO2 is going to be given off, and a reduction by NADH to form an ethanol gas. So CO2, and then it has kind of a smell to it, and you know that smell of alcohol. That's what's going to be given off. And so that's the one that you see, sorry, sitting here just talking. That's what you're going to see right here. The lactic acid fermentation we talked about in muscles is going to be here. Now, I give you some examples of microbes that would actually use these two different types. I gave you two different types as animals or humans. Fix it up. So some advantages of fermentation. It's going to be able to allow organisms to produce ATP under anaerobic conditions. So that's a good one because you might not always have oxygen there. Some disadvantages, it's not going to be very efficient in terms of capturing energy that's potentially available from those glucose molecules. Only about 2% of energy available is actually going to be captured, so that is very little. And the production potentially for toxic waste is going to be high there. So acids formed are going to help to lower the pH of the environment, and then that's going to disrupt enzymatic function. And alcohols can also coagulate cellular proteins. So coagulation is going to be where they start to stick together. So it causes these cellular proteins to stick together, and therefore they won't function properly. So now we're moving into lipid catabolism, or breaking down lipids or fats. Lipids are going to be catabolized by lipase, enzymes that are going to help to break down the glycerol fatty acid bonds via hydrolysis. And so you can see that in this picture here. So the glycerol is going to be converted to DHAP. That DHAP is going to be used as a substrate for glycolysis. And so further catabolizing, being catabolized by glycolysis, and then it's going to head on into the Krebs cycle. Now the fatty acids are going to be catabolized by beta oxidation reactions. And so with those beta reduction or beta oxidation reactions, you're going to have enzymes that are going to split those fatty acid chains into 2 carbon units and then oxidize. So that's going to form acetyl-CoA, which is going to then enter the Krebs cycle, and it's going to generate NADH and FADH2s. So remember, those are your high energy electron carriers or molecules. And so with that, it's going to help to provide the cells with energy and with carbon. Protein catabolism is going to be the breakdown of proteins. So proteins are going to be catabolized by proteases. That's going to be an enzyme that's used by prokaryotes, and it's secreted to digest large proteins outside of the cell walls. So this is outside of that organism, is how it's going to start to break those down. It's going to result in amino acids being moved into the cell, so it then moves inside once it breaks it down. It's going to then be used in anabolism to build new proteins inside that prokaryotic organism. That's going to be deaminated, or amino acid is going to be removed. And so with that deaminated to be completely catabolized during glycolysis, and or it's going to go to that Krebs cycle. And so that helps to provide that cell with energy and with carbon. So that's with your proteins. Next, we have our chemical structures and our functions here. And so with this one, we're going to go into photosynthesis for this one. And this is also going to be known as your light synthesis. This is going to be a process that allows organisms to make sugar and oxygen using light energy, carbon dioxide, and water. So those are going to be the three products that come in. And so what you're going to do, I'm going to go down towards, let's see, I'm going to try to do it towards the top this time. So I'm going to have 6 CO2s, and so that's your carbon dioxides. We're going to have 6 waters, so 6 H2Os, and then you're going to have your light energy, or your light source. So I'm just going to write light, and so those, draw our arrow, those are going to be our reactants up top. And what that's going to do is that's going to create your sugar or your glucose. So that'll be your 6 or C6H12O6 for your glucose. Sorry, O6. And then it's also going to release oxygen. And so it will release 6 breathable oxygens. And so O2 is your breathable oxygen there. So you have 6 carbon dioxides plus 6 H2Os or waters plus light. That's going to produce your products, which are going to be a glucose, which is C6H12O6, and 6 breathable oxygens. And so those are your products. Reactants up top, products here. And so here, with that chemical structure, you're going to have chlorophyll, which is going to be the organelle. That's going to have the pigment molecules that are going to be responsible for capturing energy from light. And so it's going to go through what's called photosystems. You have photosystem 1 and photosystem 2. They are going to be networks of light-absorbing chlorophyll molecules and other pigments. And so with these, they're going to absorb light energy and use what's known as a redox reaction to store the energy in ATP and NADPHs. In the thylakoid, that cellular membrane that is going to contain these photosystems is where this is going to happen. And so you see that membrane right there, that thylakoid membrane, and you see the production of that ATP and NADPH. You have what's known as the light-dependent reaction, also known as photophosphorylation or light reactions. This is where the light actually is needed. This is going to involve the reaction at the center of the chlorophyll. So you have special chlorophyll molecules that are going to be responsible for exciting electrons by way of light energy. The reactions can be either cyclical or non-cyclic. So the cyclic phosphorylation is going to involve photosystem 1. Light energy is going to excite the electrons in that photosystem 1, which is then going to pass along the electrons to the electron transport chain, and then is going to return to that photosystem 1, and the electron energy is going to create what's known as a proton gradient that generates ATP. Now, if you have a non-cyclic photophosphorylation, that's going to involve photosystem 1 and photosystem 2. So light energy is going to excite those electrons with photosystem 2 first, and then it's going to be passed to photosystem 1. Photosystem 1 then further energizes those electrons with additional light energy, and then the electrons are then going to be transferred to the electron transport chain, where they're going to be used to reduce to NAD plus or the NAD plus to NADPH. And so now the cell has to constantly replenish those electrons for photosystem 2. So it's going to do this through organic organisms, so it can obtain electrons from water, for example, with oxygenic, or it could be anoxygenic, and that's going to obtain electrons from inorganic compounds. So things like hydrogen sulfur and sulfides, things such as that. Light-independent reactions don't require light, so these are also going to be known as your dark reactions. These are going to use ATP and NADPH to generate by light-dependent that were generated by the light-dependent reactions as its energy source, and that's what's going to help it to drive their reactions. It involves what's known as the Calvin-Benson cycle, and that's going to be a three-step process in which carbon dioxide is going to be reduced to produce a molecule of glucose. And so here you see that comes in, it combines with that Rubisco, it makes your organic molecule that you see there. You use some energy to go through this Calvin cycle. It helps to create that sugar and what's known as a G3P molecule there. And then it regenerates. So this is going to be your anabolic reaction. So it's a synthesis reaction. It requires two main ATP components. The ATP is going to be generated from anaerobic respiration, fermentation, a redox reaction of photosynthesis. You'll have metabolites come in. So that'll be your second thing. It produces the precursors from which all these macromolecules and cellular structures are going to be made. And it helps to be generated from glycolysis, the Krebs cycle, and the pentose phosphate pathway. You could have what's called the amphibolic reaction, and that's going to be a metabolic reaction that can proceed towards catabolism or towards anabolism, depending on the needs of that cell. So then you can come into what's known as carbohydrate biosynthesis. And so with the carbohydrate biosynthesis, you're going to have a glyceraldehyde 3-phosphate, or that G3P that we saw right there in that circle. And it's starting to point for synthesizing sugar. So that's going to be the starting point. It's going to be used to synthesize glycogen in animals and protozoa. So glycogen is going to be the way that animals and protozoa store energy, usually, or store carbs. If you're talking about a plant, they store it as starch. So this would be the animal form of starch. You have gluconeogenesis, and that's going to be the process where the cell synthesizes sugars from amino acids, from glycerols, and from fatty acids. So it's able to make the glycogen that way. You could use lipid biosynthesis, and that's where you're going to synthesize using a variety of different routes. You can have cells polymerize, which means to have an addition or a condensation of smaller molecules of glycerol and 3 fatty acids. You can have a reverse of that catabolic reaction, and it's going to basically synthesize using a complex of polymerizations. The addition or the condensation of small molecules is what that is. Or you can have isomerizations, and those isomerizations are going to be a rearrangement of atoms, and that could be sugar and amino acids, and it'll have metabolite precursors there. Now going into amino acid biosynthesis. This is going to be synthesized from precursors of metabolites. So it's derived from glycolysis, the Krebs cycle, and pentose phosphate pathway, and other amino acids. You can have what's called amination, and that's going to be a process in which the amine group, which is the NH2, is going to form ammonia, an added precursor metabolite. Or you can have transamination. And that's going to be a reversible reaction in which the amine group is going to be transferred from one amino acid to another amino acid. You can also have what's called a nucleotide biosynthesis. That's going to be produced from precursor metabolites derived from glycolysis and the Krebs cycle. So remember, your nucleotides are going to be your DNAs, so your nucleic acids, so A, T, C's, G's, and U's. And lastly, this is going to be our integration and regulation of metabolic functions. So we're going to have 3 interactions between catabolic and anabolic reactions. So with your first one, you could have ATP is going to be released in a catabolic reaction, and it's going to be used to drive anabolic reactions. So you break it down and then you rebuild, is what that's saying. So the catabolic pathway is going to produce metabolites, which are going to be then used for substrates for anabolic reactions. So think of taking whatever molecule in that is made up of, let's say, Legos, breaking it down. You got those free Legos. Now you're able to rebuild. Now you have your amphibolic reactions, and those are going to be metabolic reactions that can proceed toward catabolism or towards anabolism, depending on the needs that that cell is going to have. Pathways of cellular metabolism can be categorized into 3 groups. So the first pathway can be a pathway that synthesizes macromolecules. So it can make things that the cell needs, like proteins, nucleic acids, polysaccharides, and lipids. Another pathway could be an intermediate pathway. That's going to synthesize metabolites for macromolecules. The third pathway is going to be a pathway that produces ATP and precursor metabolites. So this will be things such as glycolysis, Krebs cycle, pentose phosphate pathway, and the Dudorov pathway. And so with this, you're going to have regulatory mechanisms for metabolic function. And so we had 2 types of metabolic function mechanisms. You had the control of gene expression. And so basically the cell can control the amount and the timing of enzymes that are going to be produced when they need to go through these different metabolic pathways. Control of metabolic expression is also another function it has, so the cell can control the activity of enzymes that have been produced within it. And so those are going to be 2 important processes. So this is going to finish us up for our Unit 4. We will see you back with Unit 5.